From Microbe TV, this is Office Hours for Wednesday, March 19th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. I'm a virologist for 44 years. I'll tell you what I know. All truth, no lies. Welcome, everybody. Tell us where you're from. I'll give you a shout out. In the meantime, let's thank our mods for being here tonight. Les is here. I saw Vanity Nutrition here. I saw Tom Steinberg here. Thank you for joining us. Tonight we have a few things on the menu for you. We have a little discussion of what's going on in science in America, as usual. The destruction of science and public health. Just a terrible, terrible, selfish, mean effort for people. And the destruction of universities, too, because some people feel universities are elitist places. Well, fine. You destroy them all and you'll have a nation of dumb people. I do think universities teach you a lot. I went to a university. I went to several. And I teach at one. And I see what can be done. So unless you have been in it, don't tell me it's an elitist thing. I'm sorry. You just don't know what you're talking about. Okay, who's here today? A lot of your comments get cut off when I start. So sorry about that. Rita is in Idaho. I know that. Welcome. Rasul Sensei. I like being called Sensei. That's cool. Teacher, you're welcome. It's my pleasure. Wayne is in Urbana, 62F17C. What is it here? Let's see. Come on, phone. Currently, 10 degrees Celsius and cloudy. Not quite as warm as I would like it to be. Chris from Montreal, can you provide a mini lecture about TB? It's very funny. Actually, most cases I can provide mini lectures on the same day. I just saw this, though, now. TB, of course, is not my specialty. That would be, well, Michael Schmidt would be great to have on and talk about TB. But there is a TWIM, and someone gave the link later. It's coming up. Rasul is in Japan. Welcome. Will is in Changsha. My pleasure, Will, to keep you informed. Always willing to do it, happy to do it, love to do it. RFK's, Les says, RFK Jr.'s 20 years of denigrating vaccines have made measles America again. What a legacy to have of your life, that you were instrumental in reintroducing infectious diseases into the U.S., many that were gone. Kang is in Chicago, where it's 4C, raining, snowing later. Wow. Spring is behind schedule. Vanity Nutrition is having an interview Friday for medical school. Good luck. Don't be nervous. You're in control. You have a lot of science behind you. Just think of everything you know about viruses. You could steer the questions if you want. Elizabeth is in West Virginia. Abdulaziz is in KSA, where it's 23C, pretty warm. So Pete was describing, it was cut off in the chat, he walked to the Tate Modern, and I love that museum. It's just so beautiful. It was early digital and kinetic art from the 50s to pre-internet. It's got really a great collection. I'd go to London just to see the Tate. That would be excessive, though, right? Pete, yeah, here we go. Came in before. Line C in London. Walked from the Vauxhall to the Tate along the Thames. It's a great walk. It's just lovely. Hello, true listening one. What glad to have you back. Lori Loves Science 4 is in, wrong one, is in La Paz, Mexico. There you go. TWIM 267 for TB. And yes, we had the last TWIV was with Adam Ratner on measles. It's really good. We cover pretty much everything and more. So you should check it out. He's very eloquent and knowledgeable. Infectious disease physician. What do you think about 10,000 hours to become an expert? Well, so what's his name? Malcolm Gladwell said it was 30,000. Let's see. 10,000 is Malcolm Gladwell. Okay, that's cool. Let's see how long that is. That's in Outliers. How would I do that? Divided by 24 will give us the days. And that's 416 days. Divided by 365. So that would be, just a little bit over 1 year every day. That would be 10,000 hours. I think I love it. All right. I still believe in experts and expertise. This administration does not, because they have put non-experts into all the important positions. We're going to talk about one of them tonight. It's just BS to think that there are no experts and that they're not of any value, that they're elitists, right? No, they're not elitists. They happen to have studied a long time and they understand something and they can guide decisions in many different fields, not just public health, but economics and sociology and psychology and so forth. So I like the 10,000 hours. I've certainly put in 10,000 hours doing research, writing about and talking about viruses. I was told not to say I'm an expert by Lex Friedman, when I was on his podcast. He said, don't tell people call yourself an expert. People will get turned off. Okay. I don't see why. I value expertise. I really appreciate it when people learn how to do things really well, right? Like surgeons and artists and engineers and mechanics who fix your car really well. All of that. Cooks, chefs, pastry chefs, vintners. There's so much great stuff made by humans who are experts. Cheese grommet. Okay. So I like it. Rasul. Yes, Patricia. Adam covered measles. We didn't have to do a mini lecture. You should buy the book and read it. It's good. Maureen is in Northeast Ohio. Welcome, Maureen. Tom is in the Oregon Coast Range. Seems very nice there. Sandra is here. Good evening. Yes, Rita is in Southwest Idaho, where it's 13C. Mark Lamont is on the east end of Long Island. Lori, I think it's Lori, is in Pueblo, Colorado, 43F. Patricia lives less than 2 miles from the creepiest guy on earth, Ack. We all know who that is, for sure. I mean, there could be creepier people, but this is the one we all know. Yeah. Tracy is in Chapel Hill, where it's 21C. David is in Dayton, Ohio. Jessica has been missing in action in Toledo at the university. Welcome back. Jeff is in Elgin, Illinois, where it's 40F and wet. And Frank, our moderator, is here. Hello, Frank. Vista Tutti from Australia, 24C. Yeah, Australia doesn't have any H5N1. It's interesting. For now, yes. Eventually it will come in. If not by a bird, by a human, right? Yeah. Pete says destruction of climate science research is especially disturbing. Yep. Yep. All going. Sandra is in New Mexico. Oh, nice. Nice to see you. I have many virology friends in Mexico, and I have done a couple of podcasts with them over the years. Yep. Eliza is in southern Ontario, 12C and clear. Sandra says it's too warm in Mexico. I'd rather it be too warm than chilly. I prefer warm. Renzo, hello from Ontario, Canada. You've written me quite a bit, Renzo, on Instagram. I read it all. I haven't responded, but I will. Thanks for coming. Enjoy. Truth or dare? Big time on the climate crisis. Jonathan is in Encinitas, 20 degrees C, and nice. Pat is in Nova Scotia. Will is in Changsha, where it's 78. We're just going to go up to 26. That is a big jump. Wow. Because, the centigrade Celsius scale is compressed, so that's a lot. Yep. Gregory is in Bethlehem, PA, 80 miles west of Manhattan. John is in Minneapolis, 2C. Cheryl's in Santa Rosa, California, where it's 57F. Yuxing is in Hangzhou, 10C, and bright sunshine. Welcome. John is in Port Townsend, Washington, 53F and cloudy. Lagodic is 5 degrees in Chicago. Joseph is in Ontario, Canada. Tatiana Beast Mode is in Birmingham, Alabama, where it's 74F. Tractor Poodle, great name, from Bangkok. Bet it's hot there. Jeff wants to know about transmissibility. If Omicron has essentially the same spike as the original and the same pathway, droplets, could it be more transmissible? My guess is no, it's not more. Well, it has a substantially different spike, and you probably would only need one amino acid change to change transmissibility, but I don't think any good data have been presented that says it's more transmissible in people. It's viruses traveling from one person to another is more than just the virus. It's how people behave, and that's never taken into account. Elizabeth wants to rename measles Kennedy's disease. From reading Boosters by Adam Ratner, I learned that JFK, right, was a huge proponent of vaccines and passed legislation to get kids vaccinated. What happened on the way from JFK to RFK Jr.? Truth or dare is in La Jolla, where it is sunny. Stephanie is in Austin, Texas, 74F, hot and dry. Tess is in Canton, Massachusetts, 42F. $5 years? I thought I calculated it to be a year and change, but maybe I'm wrong. Yeah, Eliza, yeah, good. I'm glad you're reading it. You should. So Indiana Jones said it's not the hours, it's the mileage. It's an interesting concept, yeah. 50 weeks of 40 hours. Yeah. Now, of course, maybe you don't spend all 40 hours on one topic, right? So it stretches out. Yes, Stephanie. NIH funding and pauses is really wreaking havoc in our research labs. Yes. Some universities are having research funds withdrawn, like Columbia, $250 million, about 30% of their NIH grants. Then, of course, they're cutting the indirect costs. So either way, labs are having to stop doing research. It totally sucks that, boom, just like that, you shut it off, which is it's just really dumb. No other way to put it. It's not thought out. So people have an idea, they don't think about it, they don't look at the consequences. NIH has done great since the 1950s on making incredible products for public health. And why do you want to shut it off and do something else which is unproven? I don't even know what else we're going to do. I have a rumor that the states are going to run research, but I didn't even know that. MN it says it's a nice day outside DC. Lagotic, if you lie, you can be an expert immediately. Hey, you can say whatever you want, right? Chris wants to know if I have any opinions on Lex Friedman. I like Lex. I like the idea that he wants to talk with a lot of people. I don't like the idea that he needs all voices must be heard, like he put RFK Jr. on. It's not a voice, it's misinformation. And I don't know if that he can distinguish that. So I'm very disappointed with that because he has a big platform. And I think he should, I don't know, when you're doing interviews in so many fields, how do you know who's right and who's wrong? It's hard. Like I could tell him who to get for virology, and if he said RFK Jr., I said no, no, he's all misinformation. I would never have told him to put RFK Jr. on, or any number of other individuals. So I think it's great that he can interview a lot of people, but this idea that you have to hear both sides when one side is clearly misinformation is wrong. And also, he, I don't know if he believes in expertise, even though he's an expert himself. Kind of ridiculous. Sandra says it's important to know about a subject to talk about it. I think so, but many people do not know anything about subjects and they just blather on, as we'll see again today. We've been talking about this for months now. Truth or Dare has 20,000 hours on COVID. Began with TWIV after hearing the news. Wow, that's good. You must know a lot. Patricia's in Hustisford, Wisconsin, 0 degrees. Adam says to do 1,000 hours, you pretty much have to love the subject. Oh, I don't know. If you listen to TWIV for 20,000 hours, do we even have 20,000 hours? 2,000 a week times 52. No, 2 hours a week, or maybe 3. 150 hours a year. Oh, no, we don't have 20,000. So you must have done some other things too. Truman, 17, is in Fort Worth, Texas, where it's 55. Will says, being a complete layperson, I desperately need experts, but I have a list of over 60 experts who've made dangerous statements during the pandemic. We need to check which experts to follow. Yeah, for example, Jay Bhattacharya, who will probably be confirmed as head of NIH. He's a supposed expert who's made multiple dangerous statements. And of course, well, RFK is not an expert in anything except litigating, I guess. Tess says, in the 30s, scientists fled Europe for the U.S. Will we see the reverse? I think we will on some scale. I think many European scientists who are here now could go back to Europe. Yeah, because there are very good labs throughout Europe, and they can do that. So there will be to a certain extent, but I don't know the extent of it, right? I think of a lot of people who are born here want to stay here. Rach says, I highly recommend you get on podcast Diary of a CEO and talk about your expertise. You'll get a lot of new listeners for TWIV, and people will get to hear the facts about viruses. John says, Amy didn't trust Lex, and it took me a while to realize she was right. Amy is always right. She would call him slippery. Sandra doesn't know any virologists there. I know a bunch in Mexico City or Mexico D. I don't know what you call it anymore. Mexico DF. No, not you don't do that anymore. I don't know what the name of it is. And I know a couple of virologists, and of all places, Cuernavaca. Rasul wants to know, to become a skilled expert in virology, what subjects should one master in addition to virology? You need biochemistry, molecular biology, statistics would be great, organic chemistry would be great. I think epidemiology would be great. Ecology. These are less important, but they would be great because they really play into virology. Hello, Questella. Missed you last week. Good to see you back. Jeff says that anti-expert goes way back in American history. For example, followers of itinerant preachers in Puritan communities rejected seminary-schooled preachers. Joe is in Seattle, where it's 12 degrees C. Vanity writes, these do-your-own-research people think they're experts because they spend a couple of hours on Google. Quantifying experience is vital. Saying you have 40 years of experience studying viruses is perfect. Yeah, and then if you tell RFK, I have 40 years of experience studying viruses, he'll say, yeah, but you have a lot of conflicts of interest. And then people believe him. Whereas I don't get paid by any pharmaceutical company. None. And why Cohen is in White Plains where it's 50F. H Zoo is in Austin, Texas. Nice to see you again. We have a nice little Austin crowd here. We have an Ohio crowd. We have an Austin crowd. Mike and Lori are in Seattle, 51F. Tractor Poodle, it's 24C in Bangkok. Steve is in Kauai. All right, this is something that came up last week. David McCallum became so knowledgeable in forensic for his part in NCIS, he was also considered adding him as technical expert for the series. That's cool. And then continuing, David McCallum's big break was The Great Escape with Jill Ireland. He had a son, Val McCallum, who became Jackson Browne's lead guitarist, divorced 67. She married Charles Bronson. And let's finish up this thread. When he also acted in The Great Escape. Well, thank you. Very interesting fellow. Ducky. Sandra says, I'm interested in virology because I studied it a little bit while studying food engineering, and it seemed to be fascinating. It's totally fascinating, and of course, very relevant to foods because viruses can contaminate foods and make you sick. Gang says, are there any studies that show blood clotting factors and platelets involved in long COVID? Let's take a look. I don't know. Long COVID. Yeah, so there's they have microvascular thromboses, right? Um, and so these platelets are involved in that. Yeah. You know, you could be almost right with anything. You could be almost right with anything in long COVID. So many things seems to be awry. Yes, of course, 10,000 hours is 13 months only if you don't eat, sleep, etc. Right. It takes 5 years to get in those hours, which is not really that bad, right? Um, so you go to graduate school, which is say 4 years, and then your postdoc. It's another couple of years. You got it. Not so bad, right? Rasul said it's 5C in Nagasaki. Thank you for telling us. The French accented seal, DC 63F. The way I love viruses is because I can always lay off one day or two in the lab compared to the immunology folks. I mean, you can take a break. Whereas the immunology folks have to keep on going, yes? Nevitz Forever says most people do not know that basic and applied research findings can have wide implications across many fields of biology and medicine, for example, mRNA therapeutic vaccine against pancreatic cancer. Well, yes, most of the population does not understand how basic research contributes to public health. And, scientists are now writing opinion pieces in the New York Times and Science and Nature. Guys and gals, it's not going to do anything. You're writing to your colleagues telling them, yeah, we need research in this country. No, that's not who you want to write to. You have to write to other places where your colleagues are not. I mean, this makes them think they're doing something, right? Get an op-ed in the New York Times. Nobody's going to read it. And then an op-ed in Science or an op-ed in Nature. Nobody's going to read it except your colleagues. It's useless. Put it somewhere else. So you ask, where should I put it? Figure it out. You're a scientist. You're smart. You can do it. Oi! Paraphrase Amy. John says, I saw MIT Open Courseware, which looks completely free. There are intro to bio courses, chemistry, biochem, genetics, and more. I think biochem is really helpful to understand more virology. Yeah, MIT Open Courseware is great. Totally great. Ruth is from Brea, California, 76F. Mandrake wants to know how much university lab income comes from private versus public. I think mostly it's NIH, right? NIH, NSF, other government agencies. Their private is a small fraction of that. But, many people who have private funding are going to do better. Like if you have money from Chan Zuckerberg Foundation, they support a lot of science. You're good. They're not going to be cutting back. They should support us because they gave money to what. Yeah, Vanity says that UC Irvine has open courseware, edX, and Coursera, of course. Now, here's the problem here. Decades of research with little to show for this is just wrong. You're lying, Robert. You don't have the facts correctly. It's not true. There's a great return on investment for NIH. Sorry, you're wrong. Do you have a recommendation for virology textbooks? I want to stay brushed up on the specifics, but my professor never gave us a textbook and said most aren't good. Well, I hate to differ with your professor, but Principles of Virology, of which I'm a co-author, I admit, conflict of interest, maybe. I'd give it to you if I could. It's a great book. It goes through virology by principle. It's how I teach my course. It's a great way to learn about viruses. You can get older editions used pretty cheaply, I think. That's all you should start with, yeah. Pete says, if you don't want any science or tech, then give me your smartphone. Yeah, exactly. So Truman is referring to a column in the New York Times by a Turkish woman. I think her name is Tufekci or something, where she says that 2 of the last 4 pandemics have been started in a lab, which is just BS, right? Total BS, misinformation, wrong, lies. When she talks about how scientists in the beginning were saying, "Oh, this is a lab, blah blah blah, could be from a lab." Yeah, before they had any data. That's how scientists work. And then they got data, and they said, "Oh, it's not no evidence at all that it's from a lab." SARS-CoV-2 did not come from a lab; it came from nature. There's very clear evidence in support of a natural origin. There's zero evidence for lab origin. I know because I've read all the data and I've got 44 years of experience looking at data about viruses and making conclusions. And many other virologists make the same conclusion. Not everyone, and they're flawed. Sorry, but that's how science works also. People make mistakes. RFK Jr. stated that bird flu should spread through flocks to identify the birds that are immune. Thoughts? Well, I'm going to have a little mini lecture on that. Yes, I have a lot of thoughts. Stay tuned. Mark says, it's best to shut down research in science in general. That way, there are no facts to contradict misinformation. Just follow the principle of yell loudly and pretend you know what they're doing. Nevitz wants to know if I've seen this paper. Let's phase it. Viruses are master architects of bimolecular condensates. I have not seen it, so I wouldn't know if she would be a good guest until I've looked at it. Susan says, but the scientists aren't doing research for themselves. It's for the people, all people, not to mention the economy. So much of the U.S. excellence is initiated in academic environments. It's true. Now, of course, they will say the scientists are doing it to get rich. No scientists, very few scientists get rich. Most working scientists do not make a lot of money, and they do it for the love. They really like doing science. Tracy says, I agree with John and Amy about Lex. Let Amy know we are thinking of her. Will do. CDMX. Okay, thank you. So it's basically Ciudad de Mexico. Okay, Ciudad de Mexico. Thank you very much. Eleanor is in Ontario, 16C, melting. Christella says her baby had his first ever band concert. How old is your baby? And did they like it? Was it not too loud for their tiny ears? Oh, by the way, Markle reminds me, yeah, press like. There are 200 of you and only 90 likes. Please press like so we can get some more people in here to learn about science. Now, Tractor Boodle asks a very important question. If RFK Jr. were bitten by a rabid animal, would he just take some cod liver oil? No, 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 no. He would get rabies vaccine because he knows that would save his life. The other posturing is just to enable him to make money. I mean, during COVID, he had a party and he required everyone to be vaccinated for attendance. Yeah, it's just BS. It's just nonsense. Rach says, I've watched TWIV since 2020. Can't say I understand much of the detail, but I'm truly grateful. I've had the chance to listen to your expertise. If I hadn't, I may have been swayed by misinformation too. Well, thank you, Rach. I'm glad at least you get some benefit from it, right? Jeff says, I like Lex, but his two sides ideology doesn't work because there is only one objective reality and science provides the process to find it. Interviewing RFK Jr. will not help find it. Perfectly said, Jeff. I love it. Really nice. Yep. There's only one objective reality, and science provides the process to find it. It's beautiful. Beautiful. Les kindly inserts a link, The Principles of Virology, but you can find used versions on, your typical booksellers. Julie is in Hughesville, Pennsylvania, where it's 61F. Susan wants to know, do you know what's happening to Lipkin's funding? Stat report says it's yanked, but the NIH reporter still lists his U-54 as active. Like many other people at Columbia, it's yanked. Polka Dot Pup is in Denison, Texas, where it's 56F. Mandrake wants to know didn't Dixon help write that Monsters Inside Me TV show? I believe he was a script consultant, yeah. Yeah, yes, Michael. Just call it Mexico City or Ciudad de Mexico. Good memory. I was the one who made the comment that it's not DF anymore. Yeah, I remember you saying that. It's 26C in Mexico. Thank you, Michael. Hello, Kip. Sorry to miss you today. Kip is in New York for a bit. We'll get together again. Douglas says, asked my GP to test for the alleles HLA-B, 1505 and 401 since I've never had COVID. Just curious. Those are associated with resistance. It's interesting that you can be tested. Good for you. Mandrake says that parvoviruses can also cause platelet abnormalities. Yes, that's true. Nevitz says it's a libertarian and Ayn Rand free-for-all world now. You can believe whatever you want. You can make stuff up and make others believe it. And the R.W. SCOTUS will say it's all okay. That's right. We will get back someday. I don't know when, but we will get back. Abdul Rahman is in Brooklyn, where it's 8 degrees Celsius. Thank you, Kip, for your support of science education. It's sorely needed in these times. Izdislav says, Mexico, thumbs up. You didn't use Fahrenheit. No, I don't. U.S. is one of the few countries that uses it. I have been a scientist all my life. I use Celsius. So I use Celsius all the time. Hey, Vincent gives the real. Yeah, I don't ever mislead you intentionally or for some nefarious purpose or to make money. Never, ever, ever. Yes, Abdul Rahman talks about these scientists who write in the Times and Science and Nature. They're not writing for the public. Some of it is just vanity. It's vanity, but also they think they're doing something, right? But they're just showing their colleagues, look, I can write a op-ed in the New York Times. Useless, completely useless. Doesn't do anything. Nor writing in Science or Nature. I enjoy reading your opinions, but that's not going to make any difference. I'm already converted, right? Hello, Duncan. Long time since you've been here in North Wales. 15C. Gardening. Privet. Very Nika. Welcome back from Moscow. Pretty cold there. Pretty cold. Philip is in Wales, UK. Why do we think science is so harshly targeted today? I don't know that it's so harshly targeted. I just think that, I mean, I think the media is just reporting what the pundits are saying, and unfortunately they're wrong, right? It gets a lot of attention. I think that's the main reason. They make headlines about, this happening or this being wrong, and it gets attention. I don't think they're doing it intentionally except to get more readers. Of course, then, you have all these people who are saying stupid, non-science things, lies. And so those are reported. And the truth is not reported as much because it's not as interesting, right? So RFK Jr. says, let's let the chickens die naturally. Let's let them get all infected. Let it rip. Ay, ay, ay. So that's a headline. Yes, it's true, MN. The U.S. did have the chance to adopt the metric system, but it failed. It failed. Everybody else in the world can do it, but we can't. That's really embarrassing. Jonathan wants to know, what are your thoughts on viruses impacting disease course and cancers? Not from an oncogenic perspective, but rather, could they change the biology? Well, yeah, I think the biggest impact is on the immune system, right? So some viruses can immunosuppress and change the course of cancers, right? Measles and HIV. That's an obvious one. And of course, HIV is not, it doesn't have an oncogene like HPV, but it causes so much inflammation and the production of cytokines that cells keep dividing continuously, and that is a recipe for cancers. So lots of ways that viruses can impact cancers for sure. John is so nice. My favorite free course is right on this YouTube channel and in session for spring 2025. That would be Virology, my virology course at Columbia, which right this week is on spring break, but we've got halfway through. Halfway through. March 716 writes, when I was a clinical research nurse, I depended on FDA and NIH all the time. I could not have done my job at my small community hospital without their expertise. I think that you're absolutely right. I have no doubt. But I wonder if many people think they get along without them when in fact they don't. They don't realize what they're getting from them. Otherwise, they could be coming to NIH's defense, right? We should have rallies saying restore NIH. Susan writes, if you don't want any science or tech, make sure you never seek any health care. It's almost all a trickle down from federal funding. Lisa wants to know if H5N1 vaccine for farm chooks being developed. It's funny. It's an Australian term for a hen. Yeah, there are. But We've heard people say on Twiv why this doesn't work well because first of all, if you vaccinate a herd, not every chicken is going to respond, so there's still going to be some that are infected. So the herd will still be virus positive, and that's not good. And then you won't be able to export your chickens because people say, "Oh, you're vaccinated, but there may still be virus in there, so we don't want them." So, nevertheless, they're being developed, and maybe they'll be deployed. I don't know what's going to happen. We have a different approach, which the Department of Agriculture is looking into, and we'll talk about that in a minute. Yeah, so this opinion piece that I referenced, in which Truman 17 by Tufekci says, we were all misled, saying that 2 of the last 4 pandemics started in labs. Well, it's wrong. She says 1977 H1N1 started in a lab. No, it didn't start in a lab. It was a vaccine trial being done either in China or Russia. And the virus went from vaccinated person to vaccinated person and caused a pandemic. It's not a lab leak. When you put virus into a person's arm out in the world, it's not a lab leak. Don't be silly. And then, of course, SARS-CoV-2 did not come from a lab. Zero proof that it came from a lab. So her whole article's thesis is wrong. And fortunately, because it's in the New York Times, nobody's going to read it. That's good. SRR, thank you for your contribution to science communication. Really appreciate it. Carol, our favorite nephrologist from California, Bakersfield. Thank you for joining us. Pretty warm there, 723C. By the way, folks, hit the like button. Only 124 out of 200. It's not hard. It's right below the video window. Jeff says, your comment that scientists don't do it to get rich has the corollary that the rich do not innovate or make discoveries that solve problems. Yeah, that's right. Hiring those that do doesn't count. The rich do it to get rich, for sure. There's this funny Vanity searched Abe Books, and they have numerous copies. Of course, it's not the right way to spell my name. Maybe you'd find more. Maybe it'd be more expensive with Racaniello. I don't know. Try Racaniello. You can get them for $4. Nevitz forever. Ozempic is just one example of the results of government-funded research that has had major impact. Also, cancer immunotherapies, checkpoint inhibitors. Huge list of people being treated with modalities developed by NIH. It's just lying to say that nothing has ever come out of it. It's just pure and simple lying for whatever purpose you're interested in. So Carol says, only 3 months into 2025, and we have more measles cases than all of 24. It's sad. It is very sad. It's embarrassing. It should not happen. And I think it's a failure in public health. We're not getting vaccines to the right people. We should be working harder, but now in this administration, it's not happening. Crazy Lit says Fast Company reports that a scientist studying Gila monster venom at the VA started the ball rolling towards Ozempic. Always an interesting story, for sure. Left says, with respect to your textbook, I'll be buying the latest edition and give someone whatever version I have. Recycling will spread the knowledge. It's great, Jeff. I love that. Joe's, after vaccination, we wait 2 weeks to be effective to make antibodies, but in how many antibody generations of affinity increasing is needing. So affinity maturation keeps going on and in fact continues after the second. So it continues for months and then it starts again after the second if you have a boost, if you have a second immunization. Long time. John says, should an otherwise healthy young adult who is certain they received MMR schedule as a child get their titers checked? Well, if you got 2 doses, right, then you're okay. You don't need to. Of course, there's a possibility that you are in the 5% of people who don't respond. And you could have your titers checked and get another shot, but I'm not sure. If you don't respond, I don't know that you're going to respond then. Vanity says, it's amazing what people will freely subject their bodies to for the sake of appearing healthy. But when it comes down to actually being healthy, they'd rather eat fast food and refuse life-saving meds and vaccines. You know, did you see the other day, RFK Jr. and Sean Hannity were at a fast food joint, right? It's steak and fries. And they were saying that fast food is the new food for healthy Americans. A bunch of idiots. A bunch of idiots. Should you get a pneumococcus vaccine? Absolutely. Definitely. You don't want to get pneumonia. Hello, Airy Ozone. 62F, lovely. Christella, my house is in Celsius because I want to be more familiar with it. It's currently 19 degrees. That's the way you learn something, by using it, yeah? Bad Cat says it's 38F in Winthrop, Maine. It was 60 earlier and caught a salmon ice fishing. How cool. John says, it just occurred to me, I want to thank you all for your sterling goodbye to Dixon. I always enjoyed listening to him and his words of wisdom. We all miss him. Thank you. I'm glad you liked that episode. Tractor Poodle says, there are not 2 sides to a story when one side is lying. Stella, your child is a first grader. Any good science fair project ideas? I bet there are a lot of people here who would have ideas. Give Questella some ideas. My favorite would be to cut out a virus on a paper and tape it together and make a virus particle. It's not hard to do. You print it out, and they like doing that, and then you have a nice virus at the end. Hello, Steph SF. Our moderator is here. Thank you for joining us. James Madison is still around. Thank you for office hours. Really appreciate it. All right, I'm going to do a very quick thing on some news, and then we'll get back to your comments, and then maybe we'll have time for a mini lecture, okay? So let's turn on the screen share. I have to play this thing, yeah. Okay, this is a headline. This is a headline all over the news today and yesterday. I don't remember where this is from. Trump admin mulling over cutting HIV prevention funds in the U.S. So right now the CDC spends about $1 billion a year on HIV prevention. And those monies go to states. Where else is it going to go? For testing, surveillance, outreach. And now the Trump administration wants to get rid of this. Which is weird because in 2019, Trump actually proposed eliminating HIV in 10 years. So now he's changed his mind. This is one of the meanest things you could do. There's no reason. These people have an infectious disease. They should be treated. Why are you taking it away? Except to be mean. And I wish people would realize that it's a pure meanness action. It's disgusting. Okay, then also this week, RFK Jr. has a solution for bird flu on poultry farms. He says, let the virus rip. This is a column in the Times by Apurva Mandavili, but I'm excerpting it for you because most of you can't access the Times. So he's saying instead of killing birds, so when you find a H5N1 avian influenza virus infection in your birds, chickens or turkeys, right? You have a big farm. What you do is you kill them all. That's the current standard practice. That's what we have been doing for years. It's a terrible thing to have to do, but it's really the only solution. But RFK Jr. has a better idea because he's such a brilliant scientist. He says, when infection is discovered, farmers should consider maybe the possibility of letting it run through the flock so we can identify the birds, preserve them that are immune to it, he said on Fox News. Now, Mr. Kennedy is not a scientist. He knows no science. Zero. And so this makes no sense whatsoever. What does he base this idea on? Okay, what's wrong with it? Well, first of all, so he doesn't have jurisdiction over farms, but Brooke Rollins is the agriculture secretary, thinks it's a good idea. And she says there are some farmers out there that are willing to try this as we build the safe perimeter around the farms. Now, let me just remind you before going any further that Ms. Rollins is an attorney and a politician. She is not a scientist. She knows nothing about science. Yet she says culling puts people at the highest risk of exposure, and that's why we want to limit culling. We are concerned for you. BS. You're not concerned about anyone. You're concerned about the money that farmers make, and then they vote for you. Culling is not the solution. Strong biosecurity is. Tell me the evidence for that. So she proposes to strengthen biosecurity on farms, prevent the virus from entering, or halting its spread with stringent cleaning and use of protective gear. Really, how are you going to stringently clean an infected chicken? You're going to give it bleach? This is just BS with no scientific basis, folks. Culling is horrible, that you have to lose all that protein, but it gets it done quickly. The farmers can clean up and then they can resume production. Whereas letting it rip, what's wrong with letting it rip? Okay, so you get more chickens infected, you have more opportunities for mutation. We're trying to avoid that. And chickens would die pretty painfully if you just let the infection go through the herd. And that's really inhumane, although this administration does not care about inhumanity, clearly. It cares about money and power. Also, so the idea is you infect a population and then you see who survives, and you assume they have some genetic, they have a mutation that makes them survive, and then maybe you could identify it and introduce it into other chickens or use those chickens for breeding. That would take a while, obviously. I mean, that is a reasonable approach. So in Australia, they introduced rabbits in Australia for hunting. They got out of proportion, out of control, and they introduced a virus to kill them, the rabbits, and 0.1% of the rabbits survived. But rabbits are genetically outbred. They're genetically diverse. There is not a lot of genetic variability in chickens and turkeys. They're all pretty much the same. So you're not going to get the genetic variability you need to do this. Furthermore, I doubt that any country would want chickens from a farm where they've let H5N1 rip through it. So again, not thinking clearly. So this is not going to work. I doubt that biosecurity can even work. I don't think that you can prevent virus from getting in. Someone is going to violate the biosecurity for sure. I mean, many farms are open right now, and that's somewhat of a problem. But I think you're going to have a lot of problems by closing them, and you can't prevent it. RFK Jr., by the way, also said that wild birds don't die from avian influenza. It's wrong. He just doesn't know what he's talking about. So basically, all of this, letting it rip, biosecurity goes against standard practice without any evidence. They have an idea. They think it's going to work. For whatever reason, I don't know. There's no data to support it. Okay? All right. So I'm going to stop. We're going to get back to mini lecture, but I'm going to get back to your comments. Carol said, read an interesting study from BU who found microplastics contribute to antimicrobial resistance. That's interesting. You have to take a look at that because I wonder what the mechanism is. It could be a twin paper. Yep. Hello, Alina from Coxsackie, New York. Nice to hear from you. Gregory, I just talked about RFK's idea. There you go. Silent Runner says, vaccines for chicken will never be very cost effective. The lifetime of egg layers is only a few years. It's another problem. John says, do we know why measles vaccine is so long-lasting? And are the DNA virus vaccines all long-lasting? So the general idea is that the viruses that cause systemic infections give you longer immunity compared to viruses that cause mucosal infections. I don't know the reason for that, but if you look in my lecture, John, on adaptive responses, I have a table at the end showing the persistence of antibodies in people infected with viruses that have systemic infections versus mucosa, and the difference is striking. Dozens of years versus months. So it has nothing to do with RNA versus DNA. Now, measles virus undergoes antigenic variation, but it's not enough to provide resistance to the vaccine. We have used the same vaccine for measles virus for many years, so that's not an issue there either. It's all about systemic versus mucosal, yeah. H. Zhu asks, how does immune amnesia happen? It happens because measles virus infects B and T lymphocytes and kills them. And memory cells are lymphocytes, and so measles virus infects them and it kills them. Now, do other viruses do that? Good question. Let's look it up. Because I know measles for sure. Nothing's coming up. It may be that it hasn't been studied, but basically any virus that infects and kills a lymphocyte would do that. Joseph asks, do you think Columbia will get their funding uncut? Yeah, it seems they will. I got a letter from the university president saying they're working hard so that Columbia continue, but they do have principles. So I think they will hit a deal. I don't know what they're asking, frankly, but I think it's disgusting that the federal government can blackmail universities to doing what they want. I think that's gross. But that's where we are. So yeah, I think they will get a lot of their funding back. But remember, but also the indirect costs have been cut. That's a big problem for all universities. The NIH indirect costs. I don't know if any court action can restore those or not. But they they fool around to get around court rulings, and it's just disgusting. To achieve what? Thank you, H. Zoo, for your support of science education. Really appreciate it. Mark says, Frontline did a documentary on COVID. This is usually a good series, but they claim COVID had RNase cut sites. Well, all RNAs have RNase cut sites. Our RNAs do, all viral RNAs do. So I don't know what is the big deal, but I'll take a look, okay? Ray's Levis says, would you ever want to have the Times authors on your show? It would be nice to hear their explanation for the two different viruses found in the beginning of the pandemic. We have had Eddie Holmes on TWIV, and he's talked about these two lineages. But I do want to have Michael Warraby and his colleagues on to talk about that. We're working on that. Yeah, I like it. The French accented seal says, humans sleep, take vacations, we have sloppy leaders once in a while. Viruses don't. They keep mutating in all the alternative genetic pathways you can think of. Viruses are also resilient. Well, the viruses are way more adaptable than humans, and they outnumber them too. Yeah, Crazy Lit Asin asks, if visiting a newborn, is it wise to get an MMR booster? You don't need to, because the newborn has got antibodies, measles, mumps, rubella antibodies from the mom, and they will be protected for 6 to 8 months. So no, I would say from a scientific perspective, you don't need to do that. You could always ask your doctor. Steak and Shake that RFK and Hannity went to. I don't know if it was, I think there was fries in the title. I just don't know. Yeah, so this is interesting. I don't know who this is because there's no name, but it's whoever it is from Santa Barbara, where it's 20C, reflecting on how our global tilt towards authoritarian forms of government shows up as poor public health policies. Yeah, it's a good point. I think it's to keep your population sick so they can't do anything to combat the crap that you're pulling on them. That's what I think. I don't know why else you would do that, right? Unless you don't want to spend the money and you want to keep it for yourselves, right? And your buddies. Elena just heard that Hochschule is concerned that it's coming down here. What? Measles? Yeah, it will. It's going to be everywhere, for sure. Tom thinks this endonuclease has to do with the furin cleavage site, right? Which is a protein sequence, not a nucleic acid sequence, which just is all wrong. It's there, but it has nothing to do with being lab origin. Nope. Yeah, coloring sheets of viruses is great. I like that very much. Robin says, I've had an invite from my GP for the shingles vaccination. Do I need one after having a bout of shingles 2 years ago? Yeah, you do. You need 2 shots of Shingrix. Even if you've been infected, you should still get vaccinated. I had shingles at 55 years age, and when I turned 60 or 65, I got 2 shingles immunizations. That's the recommendation of CDC. Hello, Lise from Ohio. Welcome. Nice to see you. Angry Penguin read about a new vaccine for plants that uses small interfering RNA to protect them from viruses. So small interfering RNAs are 21 nucleotide long RNAs that are complementary to viral RNAs. So in this case, plant viral RNAs. So you would immunize the plants in a way that so they make these small interfering RNAs. And then when they're infected, those small interfering RNAs will target the viral RNA and degrade it. A cellular enzyme will degrade it. So plants use RNA in their defenses. Plants and insects do that very commonly. And so this takes advantage of that. We can't really do that because not really used much in mammals for virus defenses. Yeah, Rita says, cut HIV prevention. What could go wrong? Yeah, right. Just more AIDS cases. That's all. And more deaths. Right, Rita, he said, doesn't think it affects wild birds, which is wrong. He doesn't know what he's talking about. Just says things blatantly wrong. Let's buy eggs from Greenland, okay? Sure. Expensive. David says, my son and I did a science project on how far does a sneeze travel. We used glow germ propelled by a bellow. That's very interesting. Isn't it 100% fatal and terrible death for the bird? Isn't that why they cull? Yes, that's all right, Elizabeth. A safe perimeter? How? Pfft, they're just BSing. It's nothing. You can't do a safe perimeter. Hey. Listo says correctly, RFK Jr. believes in Lysenkoism and miasma. He's only hundreds of years behind science. Tom says Daniel Griffin was eloquent with respect to H5N1 in chickens on a recent clinical update. Oh, Costella's got a tornado warning. Good luck. Hope you're safe. It's Ohio. Rita wants to know if he even reads. That's a good question, Rita. I just don't know. I really don't know. I mean, how could he? Gets everything wrong. Patrick says there are two places I see metric done in the U.S. soda bottles, and usually there are one or two highway signs in KM, I think, to get federal funds, usually at the beginning of states at the major highways. That's funny. Uba Luby, should I shoo the bird off my porch so I don't get bird flu? This mated pair of house finches have been residents for a couple of years. Leave them alone. Just leave them alone. The likelihood they're going to give you bird flu is very low. Vanity says the chicken lifespan has me wondering, are there any germline vaccines? Would this be possible? So, Vanity, if you if you put the antigen in a germline, then as the chicken is developing, it will view it as self, and so you won't make antibody response against it. So no, you can't do that. Will, for most of my life, I knew about white blood cells being the immune protection mechanism. How do antibodies, B cells, and T cells relate to white blood cells? Well, B cells and T cells are white blood cells or lymphocytes. So, in fact, white blood cells, the word for white blood cells is leukocyte, and a subset of white blood cells are called lymphocytes, B lymphocytes and T lymphocytes. And antibodies are neither. They're molecules that are produced by B cells. Right? So B cells make antibodies. T cells kill virus-infected cells. Both B cells and T cells are considered white blood cells or lymphocytes. Okay? Angry Penguin writes, my understanding of prevention of public disease is always less expensive than treatment. Cutting HIV prevention funding makes no sense. That's absolutely right. Doesn't make any sense. It's mean and horrible. But, it's back to the Reagan era where Reagan didn't want to mention AIDS until his friend Rock Hudson died of it. Not just a gay disease, folks. Many other people get AIDS. Intravenous drug users, mother to child at birth. So anyway, it doesn't matter because anyone who gets infectious disease should be taken care of. That is called humanity, which this administration lacks pretty much 100%. Sylvia, hello again. Nice to see you. Thank you for your support of science communication. So H2 says, when you're in live chat on the phone, you don't see the like button. At least that's how it is on my phone. Maybe someone could tell H. Zhu how to do it. Maybe you have to slide something up or down. Because we're now at 171 likes out of 200, so that's pretty good. Most people are hitting it. Of course, maybe they don't have your phone, right? Hello, Sharon from Tasmania. Nice to see you again. Okay, that's Steak and Shake. Got it. He likes fries fried in beef fat. Very healthy. The thing is, he wants to make America healthy again, right? And this is crap. Boy, did I have measles? I did, Patricia. I had measles and chicken pox. And then I don't know if I got measles vaccine, actually. I did not get chicken pox vaccine. It wasn't available when I was a kid. I don't know about measles, but I got it. Yeah, I did have measles for sure. H2 wants to know what makes certain viruses oncogenic. The most common, the viruses are responsible for about 20% of human cancers. There are 11 different viruses like HIV and hepatitis C virus, hepatitis B virus, the herpes viruses, and others. So there are two general mechanisms. One is if the virus has a gene that makes the cell divide. Why would a virus do that? So small DNA viruses like human papillomaviruses, when they get into the cell, they have to use the DNA replication machinery of the cell. In fact, that's a topic of tonight's mini lecture, should I get to it. And so this protein for the papillomavirus is called E6, E7. Is produced, and then it makes the cell divide so that the virus genome can then be replicated. In some rare cases, this gene breaks away from the rest of the viral DNA and inserts into the cell's DNA and makes the cells divide continuously, leading to cancers. And other viruses, they cause chronic infections that produce cytokines all the time. Cytokines make cells divide, and that's also another way that you can get cancers. Now, why does cells dividing lead to cancer? Every time, most cells in your body are not dividing. They don't need to. Only when you need to grow some tissue, right? Why not? Well, because every time you divide, the DNA synthesis machinery makes mistakes. It makes mutations. And so if a cell keeps dividing, eventually it's going to sustain enough mutations to make it a cancer cell that it grows continuously. And that's about a dozen mutations or so. So that, in a nutshell, is how some viruses are oncogenic. Why do we have to wait to get the shingles vaccine? Well, because shingles arises after 50 years old. And so there's no need to immunize you before that. So the later you go, the greater the length of protection you're going to get. That's why. Although, some people do get shingles at a younger age, but it's very rare. GSD, Jonathan just got a dose of Shingrix. Excited not to get shingles. Shingrix is a great vaccine, highly protective against shingles. John, thank you for your support of science communication. I really liked this past week of programming on Microbe TV, and I'm contributing to keep this information kinesin going. Thank you so much. You say things so nicely. I appreciate it. RFK said there were 16 measles outbreaks last year. What constitutes an outbreak? More than one, I think, would be an outbreak. Yeah. We probably don't pick up everyone who's infected during an outbreak either, so it's always probably greater. Sylvia Pina writes, for a great insight on the Colombia situation, search for America's constitutional crisis with Catherine Frank, the Chris Hedges report. Frank's support of protests cost her her post there. Hello, Gwen. Nice to see you in Michigan. Sandra said, what's Lysenkoism? It's the idea that life arises by spontaneous generation. And there's no, inheritance. There's no one bacterium dividing to form more. It just arises spontaneously, which was disproven many years ago. Also, part of it is that if you subject an organism to some condition, let's say you make a plant thermotolerant, that will be inherited, which could be, but not always, right? Because he's saying you're modifying it not in a genetic way, but that's still inherited. Anyway, it's all nonsense. It's all disproven. Yeah, Nevitt says, imagine 30 or 40 years ago, we had no idea about small interfering RNAs, but here they are virtual everywhere. Yeah, that's government-funded research that has identified that. The ability to make monoclonal antibodies, NIH-funded research, goes on and on and on. Here's a definition from Veronika. Lysenko was a Soviet scientist and head of important science academies, and among other things, he denied basic genetic rules, which led to issues with our agriculture and repression against geneticists. That's great, yes. Nevitz says, I suspect the Drumpf cutting of HIV funding is purely anti-LGBTQ+. Yeah, totally. Did you know that in Texas they want to pass a law that being transgender is illegal? Look how screwed up this is. Let people do what they want. It doesn't harm anyone else. Oh my gosh. Hello, RH from Berkeley. Big faculty rally today responded to attacks on free speech like the arrest of Mahmoud Khalil and attacks on science, including funding for vaccines. Good to hear it. Okay, yeah, for the germline vaccine, you could make it inducible. So after the chicken is developed, then you could induce it. It's getting complicated now, all right? Too complicated. There are going to be off-target effects. I'm just worried about that. But that's how you get around the self, yeah. Oh, Silvio here. You have to click the X sign to the right of chat. Chat disappears and the like button appears. There you go. Thank you, Silvio Pina. I didn't catch shingles. I caught chickenpox as a child. Then the virus went latent in me and stayed in me. And when I started TWIV, 2008, that first year I had shingles. I don't know, I was in my 50s, I guess. R. Hurling wants to know why is so much research done using immortal cell lines? Is that the same as cancer cells? Pretty much. They divide forever, like HeLa cells taken from Henrietta Lacks' HPV tumor. They're cancer cells, and they divide forever because, as I said, they have the oncogene in them that makes them divide and divide and divide. Normally, cells divide a certain number of times, and then they die. But these, transform cells. Why? Because they're convenient. You can use the same cell line for years, and you could keep that constant as a not being a variable in your experiments. But there are downsides, of course, because they are abnormal. They don't have often the right number of chromosomes. So people use primary cell lines, which only can divide 30 times or so, and it's not as convenient, right? Like primary foreskin fibroblasts. So it depends on how much bother you want to go through. Silvio Pina understood the virus to cancer evolution. Yeah, it's all about division. I made it relatively simple, but pretty much all cancers are about cells dividing uncontrollably. And the control elements are usually oncogenes. We have in us many genes involved in regulating cell division. And those could get mutated, and then the cells will keep dividing. Boom, get a cancer. Jeff, can measles result in asymptomatic infection? I asked Adam Ratner that, and he said pretty much every infection gives you a rash. Lots of discussion of Lysenkoism. Kang says Lysenkoism is neo-Lamarckian, believing environmental adaptation influences evolution. And Veronica, eventually genetics was literally banned in the USSR, wild stuff. At the same time, we sent the first human into space. I wish we fed all our people instead of participating in the space race. I'm sorry about that. H. Zoo says, I came across a paper about bispecific antibodies neutralizing SARS-CoV-2 variants. Is this a significant treatment? it's not clear, right? Well, if you can, so first of all, these are artificially engineered, so it's not like you could induce them in a vaccine. So it would be a therapy, and so it has to be trialed. And right now, it's hard to do. There are not that many cases in one place to do a clinical trial. So I just don't know if it's clinically relevant or not. If it could treat any variant, this would be great. Because this is the problem with monoclonals and variants, is that we make a monoclonal, which is a big investment, and then it's useless. It's rendered useless by the emergence of a variant. So this would really help, but I don't know how they can be tested right now. Sophia, do you know anything about Naegleria fowleri? The brain-eating amoeba. They reside at the bottom of waters, and usually warmish waters. And when people are swimming, they stir them up from the bottom. And then if you jump in the water and the water shoots up your nose, some amoeba will go up. They will get into your brain and eat it and cause a very bad infection. So what's the solution? Well, I can't tell you not to swim in nice ponds and rivers, right? I don't know. Close your nose when you go into the water. We just can't get to 200 likes. Spamlet says Lysenko was Stalin's chief agricultural scientist and had the genetic scientists sent to the gulags. Caused many millions to die of famine in the world's best wheat-growing land in Ukraine. Hi, from Iowa City. I'm hearing from middle-aged people saying they had measles titers checked after 2 times childhood vaccines. No immunity. Maybe more prevalent than we thought, masked by herd immunity. Well, yes. So, the standard number is 5% do not respond. So I don't know why. This may be an isolated incidence. I just don't know how the study was done. But when you have high vaccination rates, there's not a lot of measles around, and that protects people, sure. So even if you haven't responded to vaccines, you're okay, right? Tractor Poodle says, are there notable recent advances in using phages to treat multi-drug-resistant bacterial infections? There are isolated examples of like heroism. The guy, Stephanie Strathd's husband, she wrote a book about this. He caught an Acinetobacter infection, multiple drug resistant, was going to die. They gave him some phages, and he's living now. But we don't have dozens and dozens or hundreds of those stories. It's still very much an active area of research. We did a TWIV in, it was in Australia last year, where 2 people summarized the status of using viruses, phages, to combat infections. You should go listen to that. That would be great. I guess it's a good thing for public health surveillance that measles makes you always sick, since that means it's always visible. Yes, it's always visible. Remember, there's a 2-week incubation period. So at the end of that 2-week period, for a few days, you can be shedding virus. So here's someone without a rash going to school or going to work or whatever and shedding virus. So it's not perfect, right? This is interesting. Visto, without Lysenko sabotaging Russian agriculture with BS, USSR could go to space and feed the people. That's a good point. And yes, less is correct. I was technical advisor for Henrietta Lack's book on HeLa cells. That's correct. Why don't we engineer our progeny to be HIV resistant using antisense? Because there are going to be off-target effects, and you're going to have other issues as well. Same thing with CRISPR. Going to have off-target effects. Yeah. Pete Glass says Lysenko is the classic case where politics destroyed science. His theory was seen by Stalin as congruent with his communism. Does this ring a bell, anyone? Vincent, correct me. COVID is not interesting anymore. Yeah, it's very interesting. Well, first of all, long COVID is very interesting to figure out what's causing it, right, and how to fix it. But the virus is also still very interesting because we really don't understand why variants emerge, what gives them a fitness advantage. We have no knowledge of that. And we'd love to know exactly how it arose from nature. Like, what was the reservoir? And, did it have to change before it entered people? I don't know if we'll know that, but that's a very interesting question. No, plenty of things to do on COVID. In fact, some basic molecular biology, too, people are working on. Kang, is Dr. Ratner correct saying measles can go the way of smallpox? Yes, he is, because there are no animals that are infected. Asymptomatic infections, well, so this is the problem. Let me get back to it. So, no animal reservoir, lifelong immunity. The problem is that asymptomatic infections cannot contribute to spread. And, those 2 days before you get a rash, you do get spread. So I don't know if that would confound eradication, because with smallpox, everyone infected got a rash and you didn't transmit until the rash came. Sophia, how does it eat the brain? I don't know specifically how it eats it. It's obviously digesting cells by secreting substances. Why the brain? Because it's forced up through the nose, right? Otherwise, it would be elsewhere. Let's see. It's a free-living amoeba, right? It's living in fresh water, single-cell organism, and it goes through your nose. And that's really the only way you get it. So otherwise, it's a benign amoeba just growing in ponds. Les says Lysenko was favored because he favored Stalin's blank slate. He did not believe in genes. Stalin's death and DNA discoveries killed off Lysenkoism. What about the mini lecture? All right, let's do a mini lecture. Let's go back to that. That will wrap us up. I did talk about viral DNA replication. All right. How do DNA genomes make more of themselves? So here's the Baltimore scheme, remember. And we have three groups of viruses with DNA genomes. And the parvoviruses, the hepatitis B viruses, and adenovirus, herpes simplex, polyoma, papillomas, all those DNAs have to be replicated to make new virus particles. And an interesting observation is that DNA replication is always delayed after infection. The virus infects the cell, but DNA replication happens late. Why is that? Well, we're going to talk about that in a moment. So how does DNA replicate? So you have a template, this black line, and an enzyme called the DNA polymerase uses a primer, which is a short piece of DNA that base pairs with the template. And that simply adds bases onto the 3' end that are complementary to the template. And in our cells, DNA replication is called semi-conservative. I don't know if you've ever heard that term, but all that means is that both strands of the DNA are copied. So here is a double-stranded DNA, and both strands are copied into double strands. So here's a picture of a DNA polymerase at the top here. We've already talked about RNA-dependent RNA polymerases. Here's the poliovirus polymerase at the bottom. It has an active site where the RNA is copied. And same thing for DNA polymerase. There's an active site where DNA is copied. One difference is that the DNA polymerases are open. The thumb and the fingers domain do not touch as they do with poliovirus. DNA is synthesized by what we call a two-metal mechanism of hold on, sorry. I lost something here. Streaming, here we go. A two-metal mechanism of polymerization where, we add bases complementary to the base on the template. Here, for example, we're adding a T, which is complementary to an A. The T comes in initially as a triphosphate, so there's a sugar, there's the base, which is T, a sugar, and then 1, 2, 3 phosphates. And there are 2 amino acids in the active site that bind metals, in this case magnesium, and they help coordinate the attacks that remove 2 phosphates and leave 1, as you can see here in this previous chain builder. And the host provides a lot. So for every DNA virus, replication always requires the virus to make at least one protein, sometimes many, and that's why it's delayed. You have to make that protein and it has to do its thing. And viruses with small genomes need more host proteins, and they may only supply one protein of their own. But viruses with larger genomes like herpes and Mpox and giant viruses, they can encode all the proteins they need for DNA synthesis and production of substrates. Really quite amazing. So the polymerase, where does that come from? Small DNA viruses, they steal it from the host. Papilloma, polyoma, parvoviruses. But the big viruses, herpes, adeno, pox, they have their own DNA polymerase. They can control it all. Other viral proteins involved. So you have a DNA polymerase, you have some accessory proteins that we'll see. You have an origin binding protein, helicases. The origin is where DNA replication starts. You have exonucleases that are needed to correct errors, and then enzymes to make the building blocks, the dNTPs that you need to make DNA. Now, we have many different kinds of viral DNAs. They're all different structures. You have single-stranded, you have double-stranded circles, and then double-stranded linear molecules of different sorts. So how do you, what one system can copy all of these? Well, there are 2 mechanisms by which all of those DNAs are copied. 2 mechanisms, a replication fork on the left and strand displacement on the right. A replication fork is exactly what it says. Both strands are replicated at the same time. You use RNA primers, so here the green are RNA primers, and eventually you have a double-stranded copy of each. And the papillomas, the polyomas, herpes, the retroviruses integrated into our genome, they all reproduce by a replication fork, as does our DNA. But other viruses replicate their DNA by strand displacement. And this requires a primer, but it's never RNA primer. It's either a protein or DNA. And it's called strand displacement because you copy one strand first and the other one is displaced. And then eventually the other strand will be copied as well. But adenoviruses and parvoviruses do that with either protein or DNA primers. So a big issue in DNA replication is what we call the 5' end problem. What is that? So you have a double-stranded DNA, like ours. You copy it. So here we're copying both strands. The red and the orange would be the new copies. And those were primed by a small piece of RNA. The RNA is then removed at the end, but then you have a gap that you can't fill in because you need a primer to fill it in. And if you put another RNA primer here, you'd have to take the RNA out and you still have a gap. So that is called the 5' end problem. And we have that problem, which is why we get old, because the ends of our chromosomes get shorter and shorter as we age until it impinges into genes that we need. But viruses have dealt with that in different ways. So let's take a look. Now, SV40 is a very cool virus, originally discovered as a contaminant of poliovirus vaccines. It was then studied extensively because it's a small double-stranded DNA, easily purified, and you could watch it be replicated in cells or in extracts, which you couldn't do for many years with eukaryotic DNA. So here's how SV40 replicates. There's a single origin of replication. And the DNA, the origin is melted, so the two strands are separated. And then both strands are copied at the same time. So it's a replication fork, and copying occurs bidirectionally. So this little bubble gets bigger and bigger and bigger until you have two double-stranded DNA products made. So here's a diagram of how that happens. So the origin is shown here. We've got leading and lagging strands on both sides. So the leading strand, you put down an RNA primer, and the polymerase can keep going. And as this bubble gets bigger and bigger, this strand just gets longer and longer. Same thing can happen on the other strand. But to the right of the ORI, you can only do lagging synthesis, right? So here we put down a primer, we make a short DNA. And as this bubble gets bigger, we can put down more primers, but there's no way to continuously synthesize DNA. So you always have these primers that have to be removed and then the gaps filled in. So that's how DNA synthesis works. And there's no end problem because there are no ends on this DNA. So that's one solution to the end problem, is simply having a circular DNA genome. And SV40 makes one protein that when it infects the cell initially, it's called T antigen. And what T antigen does is it binds to the origin. You can see 6 of them here, LT, large T, binding the origin. It melts the origin, separates the two strands, and then it binds a protein called RPA, which is a single-stranded DNA binding protein, which binds to the single strands and keeps it open. And this RPA eventually will pull in the DNA polymerase, and get this copied by the cellular DNA polymerase. So here we have the primase coming in to synthesize RNA primers, and then another polymerase comes in to make longer and longer strands. And this keeps going until both strands have been completely copied. The RNA primers are removed, and the DNA is made double-stranded. Now, at the end, you have two double-stranded circles that are intertwined, and so an enzyme called topoisomerase has to cut them so that they can separate. And also during replication, as this origin is being unwound, that results in an overwound region which would stop the progress of the polymerase. So again, another topoisomerase nicks that and relaxes these overwound regions. If only our overwoundness could be relaxed so easily. So then we have the parvoviruses, which are single-stranded DNA viruses, and their primer is their own genome. They have this interesting terminal repeat at each end of the DNA, which forms a hairpin. And the 3' end serves as a primer for DNA synthesis. And so this is a rather complicated scheme, but essentially you come in with a parvovirus genome. Here's the figure I just showed you. It is repaired by host DNA polymerase because the host does not like single-stranded DNA. So it elongates, so now you have a double-stranded copy. This can be transcribed and made into mRNA, and viral proteins can be made. And one of them will nick this double-stranded DNA at the left end. That will allow synthesis of another, one strand by strand displacement, and then the displaced strand will be copied as well. So the primer here is the DNA. So there is no end problem because the primer is the genome itself. Adenovirus DNAs are linear double-stranded DNAs. They're primed by proteins, and it's by strand displacement. There is an origin at each end, so that means the DNA polymerase binds at either end. And what happens here is the polymerase is bound to a protein called pre-terminal protein, which is linked to a C residue, and that primes DNA synthesis. And the protein remains linked to the DNA. So that first strand is copied by strand displacement. And then the second strand, it comes off, it's bound to a single-stranded DNA binding protein, which is then also recognized by the polymerase to copy it. So both strands copied by strand displacement. There's no end problem because the protein primer starts synthesis right at the end of the viral DNA. Amazing. And herpes simplex virus, a little more complicated. It's got 3 origins of replication, but encodes a lot of the proteins to do DNA synthesis. And this is interesting because when herpes viruses infect the cell, the linear DNA is converted to a circle by host enzymes. And then the circle is replicated by what's called rolling circle replication. It's nicked, the double-stranded DNA is nicked, and then DNA synthesis is initiated using the DNA as a primer, and it continues to copy this circle, making multiple unit lengths of viral genome. It's almost like a roll of toilet paper where each sheet is a viral genome. Mpox virus, the last one we'll talk about, does everything in the cytoplasm. It doesn't go in the nucleus, so it encodes all its proteins for DNA replication in the cytoplasm. And that's shown in this experiment where we're looking at infected cells. This one is stained for DNA. So you can see the nucleus, it's pox virus infected, so there's some DNA in the cytoplasm. And if you stain with an antibody to the DNA binding protein of the virus, right, which is part of the replication complex, you can see red staining in the cytoplasm, nothing in the nucleus. And the mix of the two colors shows you that pox DNA factories are in the cytoplasm. All right, I want to end up by briefly talking about DNA synthesis. As I've said before, most of our cells don't divide, and viruses do not replicate well in quiescent cells. So viruses have to induce the cells to divide so they can get host replication product, and that's done by early viral gene products, like T antigen of SV40. So remember, cells divide roughly once every 24 hours. They split into 2 by mitosis. Then they undergo a period of cell growth. They get bigger, they replicate their DNA, and then they divide. This replication is under tight control, and one of the controls is by a protein called retinoblastoma protein that controls entry into S. If conditions are not right, RB will stop the cells from dividing. And that's good because if you don't need division, you stop it. Otherwise, you're going to get cancers. And in fact, if you have mutations in this RB protein gene, you get retinoblastomas. These are tumors of kids who lack both copies of the gene. So there's no regulation of cell division, and you get tumors forming. So how do early viral proteins abrogate RB, right? They have to get rid of RB, otherwise they're never going to get the cells to divide. So here is RB. it is bound by two transcription proteins in the cell. And SV40, large T, the papillomavirus E6 or 7, adenovirus E1A protein, these all can displace RB from this complex. So here we have E2F binding this complex and displacing RB. Now, with RB bound, these proteins cannot induce transcription of genes that are needed for DNA replication and cell division. But once these viral proteins get in the cell, they pop RB off this complex. It can now bind to genes encoding cellular and some viral replication proteins. So this is how viruses make cells divide, so that they make DNA polymerase and other proteins, and they can replicate their genomes. And you can imagine that this could be a problem if these proteins stay around forever, these viral proteins. And later, when we talk about viruses causing cancers, we'll talk about how that works. Okay, so that is a very short primer on DNA replication, which I really like. I enjoy DNA replication. Okay, let's see if we have some interesting, well, they're all interesting. Jill is just getting here. Yes, my hair is slightly tousled. I just leave it. I don't really comb it very much. Yeah. Yep. And we finally broke 200 likes. Here we go. Measles has SSPE as a long-term complication. Could SARS-CoV-2 cause something similar? Sure. We just don't know, right? We have to wait and see, unfortunately. That's because with SSPE, it can take up to 10 years after measles, right? Wondering if you have any thoughts on the future of mRNA therapies. Well, I think that people are not very happy about mRNA vaccines, right? And it doesn't work for every virus, so I'm not sure what the future of those are. And as for therapies, controlling cancers and so forth, we'll see. I haven't seen any data indicated that they're going to be anything particularly good, but we need to see. We have to wait and see until the trials are done. Looks like one side is working harder in the replication fork. Yeah, the one where there's discontinuous synthesis. You got to put down a primary and make a little piece of DNA. You put down a primary and make a little, and then you have to take the RNA out and then extend the DNA. And that, yeah, it's more work on that side. Why doesn't the immune system recognize that viral DNA polymerases are suspect foreigners in the cell? That's a great question. The ones in the cytoplasm, like the pox viruses and the giant viruses, there are not a lot of those. So I think they're hidden from innate sensors. And there are sensors in the nucleus, they're for DNA anyway, but I've not heard of anything for the polymerase. It could be that they're very similar, but I'm not sure. Kang says, would the Hershey-Chase experiment work with adeno and parvoviruses? Yes, because it's the DNA that gets into cells with either virus. So if you labeled them, you would see that happening. This is great from Margot. My genetics teacher years ago said aging was the plastic cap coming off your shoelaces. This is fabulous, Margot. I love it. It's exactly what the telomeres, the ends of your chromosomes. You're born with a long telomere, and after every cell division, it gets shorter and shorter and shorter. And then when you're old, they start to impinge into your chromosomes. Yeah. All right. I think I need to wrap up. Oh, yes. By the way, Nevitz, all these mechanisms that I'm discussing took years to understand. We're still learning most of it, and it's led to therapies too, for sure. All right. I'm going to wrap this up here. Let's see if I have anyone to thank. A lot of great questions here. Gregory, thank you so much for your contribution to science communication. Really appreciate it. Thanks to all of the moderators that were here tonight. We had Vanity Nutrition. We had Tom Steinberg. We had Les. We had Steph. And is there anyone else that I am missing? I think that Frank was here, I believe. You were busy tonight. There's a lot of nonsense going on. I guess that's in a way good. It means new people are visiting. But if they just want to generate nonsense, it's not so great, I suppose. But maybe one of them will accidentally learn. Thanks, mods, for being here, taking care of everything. And thanks to all of you for joining us. Over 200 people. Our next office hours will be next Tuesday. That's the 26th of March, 8 o'clock p.m. Eastern Standard Time. I really enjoy doing this. And I love answering your questions and I love teaching you, so keep it up. Okay, everybody, have a good week. Till next week, stay safe.